Apologies for this glitch. So I am directly coming to the point, and my lab is interested in understanding the molecular mechanisms of human neurological disorders, particularly the ones which are which have no treatment available: amyotrophic lateral sclerosis, diseases caused by dysfunction in RNA binding proteins. And our goal was recently expanded by our strong collaborations with pediatric neurologists, where we started doing identification of rare neurogenetic disorders and their causative agents. So I am going to talk about a successful story today that we started in 2017-18, and then I would like to talk about our efforts, how we approach towards identifying the molecular mechanisms behind this particular syndrome. And at the end, in few minutes, if I have the chance, I would like to discuss about our efforts to identify genetic modifiers and therapeutically relevant pathways. So I am not going to spend too much time on the introduction part, but rare diseases, as Professor Thangraj said, they are not as rare as we think. There are many of them, 70 million that he was mentioning. But if you talk about globally, there are 400 million people who are affected by this condition. And this is such a devastating number, and there are no treatments available for majority of these conditions. So my journey with rare genetic disorders started when a colleague who is a pediatric neurologist at our University of Pittsburgh, she came and she knocked my door unexpectedly, and she started talking about a patient. She was following up this girl, in the front, who started showing some kind of neurodevelopmental issues. And this patient had abnormal gait, motor dysfunction, was highly ataxic, and has some kind of loss of muscle tone. So every test that was available in the textbook, we tried to do it, and this patient came out to be negative. Negative for mitochondrial dysfunction, metabolic disorder, ataxia panel, triplet repeat, anything that you can think of. So only thing we had was a patient with symptom. We labeled it undiagnosed neurological disease and no diagnosis. So that allowed us to do genome sequencing, and using that approach, we identified a variant of unknown significance, and this was leucine to proline change in one of the gene, which is called Gemin5. And this is what was title of my talk, identifying hidden gems. Because at that time, just to protect the identity of this project, we decided to name it GEM, okay, hidden gem. So parents were heterozygous, and patient was homozygous, suggesting an autosomal recessive inheritance pattern. And furthermore, we found that this patient also have evidence of cerebellar atrophy. So when we started this project, there was only one patient in hand, and no report was published, at least in public domain, to support any link between Gemin5 and any human disease. So we found it very interesting, and we started contacting people around the world to see if anybody has ever seen anything similar to this. So nothing came back for another 6 months to 8 months. But one day I found an email. I got an email from Germany, and this was patient family number 4, where this pediatric neurologist, he had 2 patients, and both patients died within 2 months of age, and they had a variant which was very close to where we identified, with similar clinical picture, with similar inheritance pattern. So we had 3 patients, and I was highly motivated to start doing functional analysis of this variant. Next month, we found another patient in France, and after that, I stopped counting, and we had significant number of patients at that time that warranted more study. And this is for students. And if you align these variants across different species, these variants are conserved, suggesting that nature, these variants must be doing something very important. That's why the nature decided to keep them even throughout thousands of years of evolution process. So we found there is evidence of cerebral atrophy, and most importantly, these variants were absent in any publicly and privately available databases. So now we decided to publish this report in Nature Communications. We went ahead and we did some functional analysis, and so that clinicians can know about this syndrome and they can start using Gemin5 as a target gene in their undiagnosed neurological disorder patients. So now these are the symptoms. And these variants that we identified, they were all over the gene, making it really difficult to do any genotype-phenotype correlation. So we initially had missense mutations that were inherited as in the autosomal recessive manner, but over time we identified patients with compound heterozygote mutations, and these patients had different degree of disease, not very homogeneous, but about 80 to 85% of patients, they had infantile onset of disease. And in 42 patients that I am talking about only 15% has age of onset after a year. Very rarely we found any patient with adult onset or who go up to adult. So when I started from Pittsburgh, we have more than 95 patients in 25 different countries around the world, and this number is increasing. Every month I get another new patient. So everybody had one patient and they thought this was not important. And when we published, I think now we are getting more and more patients. So what is Gemin5 gene? So this is a multifunctional gene that has been shown to regulate RNA metabolism as well as translational machinery. So this has been well studied protein at physiological level, where a lot of groups they did analyze the role of Gemin5 in splicing machinery. So as we know, this splicing machinery, spliceosomal complex, is made up of two components. One is RNA, one is protein. That's why people call it's a small nuclear ribonuclear protein complex, snRNP complex. So when nascent RNA is generated by RNA polymerase II, this is rapidly capped methylation. There is a methylation cap at the N-terminal part of this, and that is a signal that this need to be transported outside the nucleus. And once it is transported outside the nucleus, Gemin5 is a protein that recognizes this. And this whole recognition process is further supported by other proteins which are part of SMN protein complex, and there are multiple gem body proteins, and there are multiple RNAs which form this complex. And once this whole complex is formed, it becomes matured and it is transported inside the nucleus by another group of protein which is called important proteins. So over there, it sits down in the Cajal bodies before performing its spliceosomal functions. So this protein is mainly present in cytoplasm, but there is some evidence of this protein in the nucleus. And also this protein has been linked with regulating the different aspects of translational process. So when we started this project, this is the complex that we are talking about. One of the protein that was well studied in this complex is called spinal survival motor neuron protein, SMN protein. And loss of function mutations have been reported in this gene, and that has been linked with a disease called SMA. And there are two versions of SMA, SMN protein, which is SMN1 and SMN2, and there is only one nucleotide change in SMN1 versus SMN2. And that one nucleotide change makes SMN2 dysfunctional. And nature designed this problem, but nature gave us a hint to fix this problem for SMA patients. So recently, there are groups who designed antisense oligonucleotides that can fix this one nucleotide change in SMN2, and fixing that would increase the levels of SMN, and patients who would die within a year of age, they are going to survive, and they indeed survive. So this is a miraculous treatment that is available for SMA patients. But in our gene, Gemin5 is a part of this complex, but most of our patients, they presented with a different type of syndrome where we had motor dysfunction and cerebral atrophy. So then we decided to do further analysis of these mutations and how these mutations are causing disease symptoms in our patients. So we took blood samples from a patient who was seen at our clinic, and we isolated PBMCs. we reprogrammed them to become induced pluripotent stem cells. We took another patient who died within 2 months, and we used CRISPR-Cas9 to induce those variants in a control cell line. And these cells, they have gone through stringent quality control machine, assays. And once we were satisfied, we started asking simple questions. What is the impact of these variants on the subcellular distribution of Gemin5? So using that strategy, we found that you can see this is control iPSC neurons, and there is Gemin5, which is both cytoplasmic and the nucleus. Whereas if you have disease-causing variants, there is there's a significant decrease in the cytoplasmic expression of Gemin5 protein. So that is further supported by our immunoblot analysis, where we took these cells, we grinded them, stained them for different proteins, including Gemin5, and this is unaffected individual and from the same family. This is patient, and you can see Gemin5 protein is barely detectable. Not only Gemin5 protein, but the whole complex starts going down, meaning that when Gemin5 protein levels are reduced, other proteins, they also start going down. So this is one mutation. This is the second variant that we have. Protein is barely detectable in these iPSC neurons. And also the other complex members of SNRNP, they all go down. So then next thing that we wanted to ask, why to bother? Why to make these iPSCs? Can we do a rapid test where we can take just PBMCs and detect this kind of changes? So indeed, this is in process, but n is equal to 1, like a rare disease, we have evidence that we can detect these changes. So then next question that we wanted to ask, is this a mutation which is causing reduction in the complex proteins, or can we mimic this situation by genetically knocking down endogenous Gemin5 in any control cell line? So we took advantage of HEX cells and we depleted endogenous Gemin5. And doing so, we found that other components of this whole complex, they started going down. So it appears to us like these variants are causing the dysfunction by a potential loss of function mechanism, where Gemin5 protein is not available and it's not there to perform its function. So what are the downstream consequences? In order to do that, we decided to do in vitro assembly assay, where we labeled one of the components of this complex, SNRP complex, and then we took cytoplasmic lysates from iPSC neurons from control as well as from patients, and we incubated them to see how reliably this complex is formed. So using that approach, we found that there is indeed, this is control, these are patients' cells, and there is indeed a reduction in the complex formation. And not only that, when we did biochemical interaction studies, we found that these mutants, they have limited ability, or their ability to bind to other components is significantly reduced, which you can appreciate over here in the last lane. So then now we have a new syndrome which is caused by dysfunction in Gemin5 gene. And while we were studying Gemin5, when we published it, other groups started contacting us with newer rare genetic disorders with hope that we can help them in identifying the mechanism of these variants. So we got another syndrome which is called Gemin4 syndrome. And all of these patients with Gemin4 variants, they had symptoms with everything that we have seen in Gemin5, but also they had skeletal abnormalities, suggesting that there is something else is going on, which is now part of our ongoing study to understand the molecular mechanisms. But so far, what I talk to you is in vitro data or patient data. What is the relevance of these findings to animal model system or in vivo model system? Is there any way we can identify some pathways, or we can identify some targets that can be potentially modulated by using any approach, and that can be used to develop a therapeutic intervention? So in order to do that, we started using Drosophila as a model system, which is one of our expertise, and we use Drosophila as a complementary model system to iPSC neurons, mouse model, as well as a neuropathologist who is present at our university. So Drosophila model has many advantages, and many fundamental biological pathways were discovered in Drosophila, including sleep cycle. Few years ago, somebody was given a Nobel Prize who identified this circadian rhythm in Drosophila. So you can see if you take males and females, put them together, they go through different developmental stages. And this developmental paradigm is fully uncovered, meaning that we know every minute of their developmental paradigm. So this can be potentially used as a model organism system for studying neurodevelopmental disorders. Gemin5 is conserved in Drosophila, and it is called rigor mortis. So we took loss of function alleles of rigor mortis, and we did the same experiment that I showed you before, where this is control, and these animals, they go through different developmental stages. But if you reduce the levels of endogenous rigor mortis, which is human Gemin5, there are two observations that we made. Number one, these animals, they have developmental arrest. They take too long from going from one stage to another stage. Second is they die in late pupal stage. Very few escapers we got. So this was an interesting observation as a geneticist, because we can use it as a readout for identifying another gene that can bypass loss of function in Gemin5. So not only developmental defect, we have evidence that these animals have motor dysfunction, and they have reduced lifespan. So in order to identify genetic modifiers, we use the power of our Drosophila model system, and we started modulating genes that are part of this SNRP complex, which you can see in the middle of this slide. So since the complex is conserved, we started taking alleles where that can overexpress or that can deplete endogenous levels of these targets. So after doing a series of genetic screen experiments, we found this. So this is control. This is rigor mortis RNAi, where animals die in late pupal stage. When you overexpress SMA in SMN protein in these animals, there is a black and white change, meaning that these animals, they start surviving. So that was a big breakthrough for us. And so question that came up, whether it is only happening in Drosophila or it is true in human patients. So we quickly decided to go in human related cells or human patients. And just to give you a little bit more background about what we were trying to do is, there is therapies available, or there are drugs available that can upregulate the levels of SMN protein. And this has been used in human patients and have been somewhat effective. So these drugs can be antisense oligonucleotides that can fix one nucleotide in exon 7 of SMN2, or there are some small molecules available. So we asked this question: what is the relationship between Gemin5 and SMN overexpression? So just forget about mutation. So we took control cells and we started upregulating and SMN genetically. And you can see when you upregulate SMN in control cells, there is increase in Gemin5 levels. So when you upregulate SMN, Gemin5 goes one. When you have mutations in Gemin5, Gemin5 goes down, SMN also starts going down. So it appears like these two proteins have some kind of equilibrium or a relationship that each proteins are tightly regulated. Then we got antisense oligonucleotides, which have been used in human patients, and we treated these cells. These are just control ASOs, and then we used different doses of ASOs, and you can see there is a dose-dependent increase in Gemin5 level in these cells. So now next thing that we wanted to do, I was presenting actually this data in a meeting in Vienna, and there was an SMA expert sitting in the audience, and she asked me that you are describing a new syndrome. What happens to the Gemin5 level in SMA patients? So after an hour of conversation, I convinced her to do that experiment. So she, my collaborator, Dr. Bruni Held, she had iPSC neurons from type 1 patient, which is the most severe, type 2 and type 3, and she looked for the levels of Gemin5 in these cells. So this is type 1 SMA patient. You can see in the panel here, there is a reduction in SMN protein, and also Gemin5 goes down. Similarly in type 2, when you have reduction in SMN, Gemin5 starts going down. So just opposite happens, suggesting that these two proteins are tightly regulated. Now we went to our patient cells, and as I showed you before, our Gemin5 protein is barely detectable. When you upregulate, genetically upregulate SMN in these cells, there is some expression of Gemin5 protein. So suggesting that if you upregulate endogenous SMN, Gemin5 protein also starts at least expressing. So similarly, we tested in another mutant cell lines, and then we used ASOs that have been used in human patient cells. And we found exactly similar things, that when you upregulate, treat these cells with ASOs, there is increase in SMN, but also there is an increase in Gemin5 levels. So these findings were very interesting, and now we are moving towards testing these things either in rodent models, or we are trying to give this option to the clinician whether they want to try a non-invasive method, which is a small molecule or oral drug that they want to try in human patients, if anybody is interested. But while doing this, we decided to develop mouse models, and we decided to knock out endogenous Gemin5 to generate this mice. And this was one of the failed projects that I proudly say, because when we knocked out one allele, the mice was completely normal, similar to human patients, but human heterozygote individuals. But when we tried to make homozygous by making complete null, this was embryonic lethal. So that was a project that we initially started in mice, but then quickly we moved to another approach where we use CRISPR-Cas9 to introduce these variants in mouse Gemin5. And again, we created these point mutations that we identified in human patients, and heterozygotes were completely normal, as we have seen in human patients. But when we tried to make them homozygous, they were embryonic lethal. So another 2 years were gone. But good news is we made a conditional mice quickly, and now this conditional mice, which is being rigorously characterized, and our preliminary data shows that these mice have some evidence of developmental abnormalities, as well as these animals have some evidence of abnormal gait, which is one of the features, clinical features of human disease. And I wanted to just quickly show you this video before I end. And so this is control. Somehow, yes. So this is Gemin5 mutant mice, and it doesn't look similar to controls, meaning that there is some underlying problem which we are trying to document by using MRI scan and then other types of behavioral assays. It looks like this mice has some kind of motor dysfunction as well. So with this, I would like to summarize what I talked so far. We identified a new syndrome, and it is likely to be potentially loss of function mechanism, and we identified a genetic modifier that can be modulated by using either genetic approaches or by using small molecule approaches. And now we are further looking more into the mechanism on one hand. On the other hand, we want to offer whatever we learned about Gemin5 to our patients. And one of the patient, mom, she recently started a foundation to support research, which is called Gemin5.org. And we are going to have a Gemin5 day in University of Pittsburgh to tell them what we have been doing. And I'm not going to read my summary part, but I am blessed to have collaborators in 25 different countries who have been instrumental in helping us in reaching up to this stage. And this is my team. And I'm very grateful to the NIH for supporting our project. And thank you very much for your patience. I'm ready to take any questions.
One quick question, please. Is induced by which? Induced by which?
Sorry?
Conditional mice.
Oh, tamoxifen inducible. uh, have been very happy with my flies and iPSC systems, but there was a need to develop these mice because we want to test those, SMN overexpression in these animals. These are tamoxifen inducible animals. I have a full-fledged presentation in case if you want to talk about the mouse model. We'll be happy to discuss this.
So for the paucity of time, we will end here, but Kiran will summarize the session here about presentation. Thank you. Thank you all. So we had a couple of wonderful talks along with the technical pitch, but our team was successful to have a talk of Professor Udai Bhan Pandey also. Also, thanks to Dr. Conrad for keeping us engaged with his answer session and the audience in that period. So, just to summarize the talk, we had a great session from Dr. Dev Conrad about on the talk population genomics for health. The talk concentrated on human populations of India, representing Indian population graph and showing us the genetic evidence for recent population mixture in India. Population with high IPD score, which was a extensive research what they performed in this work, shows us